Hi, I'm Coby Viner, a PhD student at the University of Toronto, and today I'll be talking about modeling methyl-sensitive transcription factor motifs with an expanded epigenetic alphabet. Just as different transcription factors distinguish between different nucleobases, some transcription factors are known to distinguish between different modified nucleobases, such as many shown here. As you can see, a lot of different transcription factors have distinct methylated motifs versus their unmethylated counterparts and can be sensitive to a variety of different epigenetic modifications as shown on the left. Work in this area has shown that a number of different transcription factors across a variety of families have particularized sensitivities to methyl-sensitive transcription factor motifs, as well as to various combined changes in epigenetic transcription factor binding activity. This work has focused on epigenetic modifications to cytosine, including the widely known 5-methylcytosine, but also its downstream oxidized modifications, including 5-hydroxymethylcytosine, 5-formylcytosine, and 5-carboxylcytosine, as shown here. These form the basis of the active demethylation cascade through which 5-carboxylcytosine is thought to often be converted back through the action of thymine DNA glycosylase, followed by base excision repair, to unmodified cytosine residues, although we now know that there are other mechanisms through which this can occur. We sought to expand the standard ACGT DNA alphabet to directly account for these different epigenetic cytosine modifications and allow us to model them. To do so, we added M to represent 5-methylcytosine, H to represent 5-hydroxymethylcytosine, and so on. Because of the double-stranded nature of the DNA double helix, and in order to retain information, we also added symbols to represent the complementary base pairs of different modified cytosine residues. These included 1 to represent a guanine that's complementary to a 5-methylcytosine, a 2 to represent a guanine complementary to 5-hydroxymethylcytosine, and so on. Using these various symbols, we can now directly encode them into our bioinformatics workflows to allow us to model these, but we also need symbols to encode ambiguities, or cases in which we're uncertain about the underlying epigenetic modification. Often, during conventional whole genome bisulfite sequencing, you cannot distinguish between a 5-methylcytosine and its hydroxymethylated counterpart. To represent this, we use an X to represent either M or H, and a 7 to represent a guanine complementary to 5-MC or 5-HMC, that is a 1 or a 2. Similarly, for unknown cytosine modification states, such as from conventional sequencing, we use Z to represent any cytosine, and a 9 to represent a guanine complementary to a cytosine with unknown modification state. Leveraging this expanded epigenetic alphabet, we were able to adapt the entirety of the meme suite, some of whose tools are shown here, in order to represent these different transcription factor modifications and model them in terms of de novo motif discovery, as well as downstream analyses through the various tools shown here, with a particular focus on Centrimo, where we use central motif enrichment as an assessment for direct DNA binding. Centrimo outputs scores for each motif that are representative of their probability of likelihood in terms of their central binding. For example, for a standard E-box motif pertaining here to the motif CMYC, CACGTG, we might get this p-value 3.7 times 10 to the negative 9, or around negative 19 in log space. We can compare that score with a hypothesis of a particular modification of this transcription factor binding site, such as what happens if the central CPG is hemimethylated. That is, the central cytosine is transformed to a 5-methylcytosine, represented here by the red M. We can then compare that score of negative 6 to the unmodified score and compute the difference of these p-values in log space, which results in this score being positive for a preference toward the modified state, that is a preference for M or H, and negative for a preference for the unmodified nucleobase. So if we perform this subtraction here, we get this value of negative 13, which shows that in this case, the unmodified motif is quite strongly preferred with respect to its modified counterpart. And so we could then plot this particular hypothesis here at a given threshold of cytosine modifications, where you use a threshold to call these different modifications genome-wide. And then we can ask, well, what happens with all the different hypotheses for every modification to the central CPG? So what if it's hemimethylated on an opposite strand, or completely methylated, or hydroxymethylated? And we can see here that all these values are below 0, which is exactly what we would expect, given that CMYC has widely known to have a strong preference to bind in an unmodified context. And now what we can do is take that same point at that same position of -13 at this 0.7 threshold, and looking at all of these different hypotheses, we can now flip our axis. So instead of plotting that score of -13 on the y-axis, we're going to flip it to the x-axis to allow us to accommodate different transcription factors. And so by doing that, we expand this plot to what's shown here, where that single value of -13 is this small dot just to the left of this red line representing 0 in a log scale. So even small departures from 0 are quite substantial. And we can then add those hypotheses from before, and you can see again, they're all below 0. But now we can expand this to a wide variety of different transcription factors across ENCODE. And we can see, for instance, that there's a number of different transcription factors with strong preferences, both for and against 5-methylcytosine or 5-hydroxymethylcytosine. And as some examples, CTCF and its associated proteins have very strong preferences below 0, representing their strong preference to bind only in unmodified DNA contexts. This is something that's been quite widely known, but you can see that, for instance, there are some motifs that do appear to have a preference to bind even for CTCF in a modified context. So now what we want to do is go back to this notion of modified motifs being distinct from their unmodified counterparts and see if we can establish a kind of clustering, as was shown in this initial study in eLife, back when this work was first becoming popular. And so to do that, we leveraged a tool called RSAT matrix clustering in order to cluster these different motifs in a hierarchical clustering method to group similar motifs together. So what that looks like here is we can combine, for instance, these similar motifs and get combined root motifs for each. And so going through this, we might, for instance, see this motif as the root motif of that particular cluster. And we can do this across all of our different transcription factor binding sites that we assessed. We sought to combine those previous clustered motif preferences with our prior scoring methods. To do so, we developed a visualization that shows these clusters and their scores together. Here, for instance, we see a group of 2 motifs that were clustered together, both with strong preferences toward unmodified binding, as shown in different shades of blue. Similarly, here we have a group of 2 separate motifs, each in their own clusters, one with a strong preference to bind in an unmodified context, and the other in red in a modified context. And similarly, we can expand this out for all the different motifs for this transcription factor, as well as obtain root motifs for each cluster, such as the one shown for the cluster on the bottom. By combining these, we can, for instance, look at particular transcription factors, such as here for CBP beta, and look at different groups of motifs with each of these particular scores. Here we see one group with a strong score for unmodified binding, as shown on the bottom, and on the red, a strong score for binding in a modified context. One of our main results was for OCT4. In particular, the highlighted group of transcription factor binding sites was found to all have a strong preference to bind in a modified context, something that was previously unknown. We sought to validate this using cut and run, followed by different library preparations to sequence in both a conventional, methylated, and hydroxymethylation specific manners in order to validate these preferences. In doing so, we found that our predictions were correct, and we found strong results for hydroxymethylated motifs in the hydroxymethylation specific sequencing, as compared, for instance, with the unmodified motifs or with the whole genome bisulfite sequencing, which had similar but less strong preferences for the methylated motif with respect to the unmodified sequencing. And so that leaves us with our open questions, of which we have quite a few. For instance, how do these scores and our clusters combine in the best manner? Is it the case that scores that are very close to 0 really represent a motif with an equivocal preference? That is, one that might genuinely be able to bind in both modified or unmodified contexts. And how locus specific are these preferences? Does this have to do with the purity of our clusters? And are there particular metrics that might be really good at measuring this versus others? And could we devise some new metric that allows us to quantify this in a manner that's generalizable across all of our transcription factors? And with that, I'd like to end by acknowledging the many people that were involved in this project, especially my supervisor, Michael Hoffman, as well as our many collaborators in different labs across the world and our funding. And thank you for your kind attention.